DNA is often referred to as the building blocks of life, but what does the word DNA actually mean? The word DNA is actually an acronym for deoxyribonucleic acid. DNA is a polymer. A polymer is a large molecule made up of repeating subunits. In the case of DNA, these subunits are called nucleotides. A nucleotide is a molecule made up of three chemical groups: a phosphate group, a sugar group in the form of deoxyribose, and a nitrogenous base, of which there are 4 types. When many nucleotides are joined together to form a polymer, they are connected together by alternating phosphate and sugar groups, forming the backbone of the DNA. The nitrogenous bases protrude outwards and are free to form base pairs with another DNA molecule. DNA is composed of 4 types of nucleotides, which differ only in the chemical makeup of its nitrogenous base. The 4 types of nucleotides are known as adenine, cytosine, guanine, and thymidine, represented as the letters A, C, G, and T, respectively. These 4 nucleotides are categorized in 2 groups known as purines, which contain a double ring, and pyrimidines, which contain a single ring. Generally, a purine base pairs with a pyrimidine base, where A pairs with a T and a G pairs with a C. These base pairs are held together by weak bonds known as hydrogen bonds, which are different from strong chemical bonds. When 2 single-stranded molecules of DNA containing complementary sequences pair up, they form double-stranded DNA. DNA has directionality, or polarity. Notice the ordering of the alternating phosphate-sugar backbone. The end that contains the phosphate is known as the 5' end, and the end that contains the sugar group is known as the 3' end. Double-stranded DNA is bidirectional, also referred to as antiparallel. When 2 single strands base pair, the 5' end and 3' ends of adjacent strands point in opposite directions. The 3' end of DNA is of particular importance during DNA replication. The enzymes involved in DNA replication require the presence of a 3' end in order for DNA replication to proceed. During the replication process, one strand can be used as a template to make a copy of the other strand through complementary base pairing. Double-stranded DNA resembles a ladder, with the phosphate-sugar backbone forming the sides of the ladder, and the base pairs forming the rungs of the ladder. This ladder winds up to form the secondary structure of DNA. The secondary structure of DNA is known as a double helix, with the 2 strands winding around each other. This helical structure is maintained by the stacking of the base pairs. The ring structure of the nitrogenous bases is planar, or flat, which allows for this stacking of the bases. The base pairs point towards the center of the helix, and the phosphate-sugar backbone points outwards. Spaces between the phosphate-sugar backbone forms 2 main grooves, the major groove and minor groove. The helix does 1 complete turn every 10 base pairs. When viewing a DNA sequence in the form of a text file on a computer, usually only the sequence from one of the strands is displayed. The left side of the sequence corresponds to the 5' end, and the right side of the sequence corresponds to the 3' end. Reading the DNA sequence from left to right is equivalent to reading it from the 5' end to the 3' end. One strand is all that is needed to study the DNA sequence on a computer, because a computer program can be easily made to display the complementary strand by using the displayed sequence. Remember, A pairs with a T, and G pairs with a C, and that the complementary strands are antiparallel.